Okay, hello everyone. So this is the third in our seminar series, and in some senses, I've always wondered whether I should give this lecture last or whether I should give it now. And the reason I'm giving it now is we are going to move away from energy balance, and where we covered calorimetry, metabolic rate, energy balance, and food intake in the past two lectures. And those lectures were not lectures about biology; they were physics lectures. So we're now going to move into another variable we extract from calorimetry, which is the respiratory quotient. And so the first half of this lecture is pretty much going to be chemistry. And then finally, we're going to move into a bit of biology after 2.5 lectures of an animal phenotyping course. So the talk is entitled, so this is a reference to a film from, I think, the 1960s by Stanley Kubrick. This is a reference to the film Dr. Strangelove, and the subtitle of that is Using Calorimetry to Study Carbohydrate Metabolism, or How I Stopped Caring About Energy Expenditure and Learned to Love RQ. So what we're going to do is talk today. First of all, we're going to talk about what respiratory quotient, known as RQ, or also known as respiratory exchange ratio. I think I fixed it, so I use respiratory quotient at all times in this talk, but it is possible there's a few RERs in there. If you see RER, just consider it equivalent to RQ. What is it in metabolic terms? What is this value? And then we're going to talk about what does your respiratory quotient over a prolonged period of time, and in this case, we're going to think about prolonged period of time as a full circadian cycle, 24 hours, tell us physiologically. Then we're also going to talk about what does the range of respiratory quotient over a 24-hour period tell us. And so this is more about how much the RQ has in terms of amplitude, because it's not static over those 24 hours. It varies quite a lot. And then finally, we're going to move that into a more biological setting and think about what and how RQ can inform us about carbohydrate and lipid metabolism in mice. And we think those things are important for understanding how obesity leads to metabolic diseases such as diabetes. So I'm going to start with this slide, which is what is metabolism? And I think we should all know it, but when I actually came to give some lectures to undergraduates, I found myself actually looking this up. And so metabolism is the set of life-sustaining chemical reactions in organisms. And the three main purposes of metabolism are the conversion of food to energy to run cellular processes, so essentially to make energy from food, the conversion of food or fuel to building blocks for proteins, lipids, nucleic acids, and some carbohydrates. So that's growth, is another way of thinking of it. And then finally, the elimination of nitrogenous waste, because we can eliminate water and carbon dioxide relatively easy, but nitrogen, we have a whole separate system necessary for eliminating nitrogen. Now, this definition comes from a highly respected source. This is the Wikipedia definition of metabolism, so always a good friend to the scientist. But why I mention this right now is that RQ is a compound variable and it can be impacted by all aspects of metabolism. So this means it's potentially incredibly powerful because it can tell us about all these different things, but it also means it's very difficult and you need to have a lot of care when you interpret changes in respiratory quotient because they can be caused by so many different things. So we had these slides before, but I'm going to recap. So respiratory quotient is measured by indirect calorimetry. So we normally think of indirect calorimetry as a device for measuring energy expenditure, which we calculate from oxygen consumption and carbon dioxide production. And carbon dioxide production is not in a one-to-one ratio with oxygen consumption. The amount of CO2 produced varies dependent on the source of energy, whether it be carbohydrate, fat, or protein. And dividing the amount of carbon dioxide produced by the amount of oxygen consumed gives us a value called the respiratory quotient, which provides some information about fuel usage. So a bit more on RQ. RQ is VCO2 divided by VO2. Now, burning carbohydrate has an RQ of 1 because we have in a molecule of carbohydrate, and for example, glucose is made up of 6 of these in a ring structure, we have a carbon and a water, so carbohydrate. Now, if we want to oxidize this fully, then we're going to need 1 molecule of oxygen in order to convert this carbohydrate into a molecule of CO2 and a molecule of water. So we have a 1-to-1 ratio between CO2 and oxygen. Conversely, burning fat has an RQ of 0.7 because this is a basic unit of fat, a carbon bonded to two hydrogens, and we find these in long chains of carbons with a carbon backbone, usually 16 to 18 carbons long for most of the fuel fatty acids. And to fully oxidize this to a molecule of carbon dioxide and a molecule of water, we need 1.5 molecules of oxygen, 1.5 molecules of O2. So therefore, we get a ratio of 1 divided by 1.5, which comes out as about 0.7, because each of the fatty acids we are oxidizing contains a carboxyl group, which has 2 oxygens in it. So that drives it a bit above the pure mathematical 0.66. So how do changes in metabolism impact on respiratory quotient? So in general, I would argue people really think about respiratory quotient in terms of the oxidative coefficients. So we've seen 2 of them. Carbohydrate is 1, lipid is 0.7, protein is 0.9-ish because it depends on the amino acid. And as not all proteins have equivalent amino acid compositions, different proteins will have slightly different RQs when you oxidize them. However, it is the case that processes which indirectly affect oxidation will impact on respiratory quotient. So this is where we begin to see so many different processes able to affect respiratory quotient. So for example, lipolysis, the process of breaking down triglyceride to release fatty acids, if we inhibit that, we will most likely impact on respiratory quotient, because if we have no fat available because we've prevented lipolysis to burn, then by default, carbs will be a greater proportion. Even if we don't change the amount of carbs being oxidized, carbs will be a greater proportion of the oxidative metabolism going on. And as such, RQ is a unitless variable. It's a ratio. We will get a higher RQ because we're doing more glucose oxidation and less lipid oxidation as a proportion of that ratio. Okay? So blocking lipolysis will potentially increase respiratory quotient. And so virtually every metabolic process will impact on nutrient usage or fuel supply to a greater or lesser extent. However, we end up with this single compound variable, respiratory quotient. So how do we interpret it? And, well, let's consider some of the metabolic processes first that affect respiratory quotient in an example organ. And the example organ I've chosen is brown adipose tissue because it does almost everything. And that means it's quite a nice one to look at. So initially, people thought brown adipose tissue was very heavily dependent on lipid oxidation. It certainly does a lot of that. Subsequently, it was discovered it also takes up huge quantities of glucose and oxidizes those. And also, really quite recently, people have started to think it may be a major location of protein oxidation and amino acid oxidation. In addition to these catabolic processes, it has been known for decades that brown fat has extremely high rates of lipid biosynthesis, so it's also a powerfully anabolic organ. Okay, so we can think about brown fat and what goes on in brown fat. So glucose, we've got glucose transport, it can go through glycolysis, and then it can ultimately end up at pyruvate, and there are 2 fates for the pyruvate. The majority of pyruvate in bats seems to be dumped out as lactate. However, it can also go for oxidation by the citric acid cycle. That will then produce NADPH and FADH, NADH and FADH, which will be used by the electron transport chain, and in theory could ultimately end up as ATP. However, in brown fat, there is a short circuit, so we actually see most of this energy that's being produced by the TCA cycle end up being used for the production of heat via the enzyme UCP1. In addition to oxidation in the TCA cycle, we can also get export of citrate, and this will ultimately be used by fatty acid synthase to make lipids. We need to replenish the TCA cycle if we take citrate out, so we use pyruvate carboxylase for that. In addition to glucose metabolism, we have fat metabolism, and brown fat takes up huge amounts of lipid. It hydrolyzes lipoproteins via the enzyme LPL. This releases free fatty acids. These may go directly for oxidation, but there are some suggestions they have to go through a storage process first. And then these fatty acids can be liberated by lipolysis, where they will go for beta oxidation. And again, we run through the same processes. Okay. Now, all of these processes will impact on respiratory quotient. And what I'm going to do now is I'm going to talk through the processes and the changes in these processes, which will drive an increase in respiratory quotient. So given that glucose oxidation leads to a high respiratory quotient and fat oxidation leads to a low respiratory quotient, it's not surprising that if you just increase glucose oxidation, you will increase your respiratory quotient. Equally, decreasing your fatty acid oxidation will also increase your respiratory quotient. A decrease in fatty acid transport, so there's less fat coming into cells, can potentially also lead to an increase in respiratory quotient because there's less fuel for oxidation. Therefore, glucose oxidation becomes a larger proportion. Decrease in lipolysis, we've already mentioned this. Possibly an increase in fatty acid esterification, again, locking up fatty acids and stopping them being used for oxidation. And finally, an increase in fatty acid synthesis from glucose. Fatty acid synthesis has an RQ of 5, converting glucose into fat. And although it's usually a relatively small proportion of the mouse's metabolism, it can drive RQs up to quite high levels. And there's another complexity to this concept of different processes interacting. So we've already talked about this mathematical concept that if you reduce your lipid oxidation, even without changing your glucose oxidation, because you're working on a ratio, glucose becomes a larger part of your pie chart and your RQ goes up. However, metabolism in mice is largely a zero-sum game. Mice are small, they have very high requirements to produce heat, and the amount of energy needed to heat a mouse is proportionate to its body weight. We covered these in the last 2 lectures. And if a mouse stops expending energy, then it will drop its core temperature by 1 degree C every about 4 minutes at room temperature, and thus become hypothermic and die. So if you drop your fatty acid oxidation, you need to replace it with something else. So if mice must expend energy proportionate to body weight, you can't just reduce your lipid oxidation. It has to be a reduction in lipid oxidation that is matched by other metabolic processes. So these processes definitely have this interconnectedness. So in terms of the ranges of RQ, RQ is often quoted as ranging from 0.7 to 1, and that's from pure carbohydrate oxidation to pure lipid oxidation. We discussed why those 2 values exist. However, the actual range of RQ is somewhat wider. And so the first one I mentioned is lipid synthesis. It has an RQ of 5, and that can practically drive RQ to 1.1, 1.2, though someone told me the other day they'd seen 1.3 reliably in animals when you get, if you can generate a state with really, really high lipid biosynthetic rates. Now, one important point here is this is RQ is 5, but the net value of turning carbohydrate to lipid, then oxidizing the lipid, is actually still 1, as though you just oxidized the carbs in the first place, because you do a small amount of 5 to go from your carbs to your lipid, and then quite a lot of 0.7, which all balances it back down to 1. Okay. There is another state which can drive RQ below 0.7. Well, there are actually technically 2 states. One is relevant for us as diabetes researchers, and that is ketone production. If you produce ketones, that has an RQ of 0. Now, they're often produced from fat. That RQ will be paid back in much the same way as lipid biosynthesis, the example I just gave. However, if you lose your ketones in urine, which can happen in diabetic animals, you can drive the RQ as low as 0.65, maybe even a bit lower in type 1 diabetics. The other state that will drive your RQ below 0.7 is heavy consumption of alcohol, because that has an RQ of, I think, 0.66 to oxidize alcohol. Less relevant for us in the work we do. Okay, so if we've measured our RQ in our mice over a 24-hour period and we've averaged it over that period, what does this mean? What does this value mean? So our wild types have an RQ here, our knockouts, it's a bit lower. Does this mean we've changed our knockout to be more reliant on fat? So I'm going to argue that's not the case, and I'm going to argue it cannot be the case. So the hypothesis is a change in this 24-hour average RQ cannot be explained by any genetic alteration or drug treatment that only affects either carbohydrate or lipid metabolism. So it doesn't matter how heavily you nuke beta oxidation, for example, you're not going to change your average RQ over the circadian cycle, certainly not in the long run. So why is this? So what do the RQ values measured over 24 hours mean physiologically? So we thought about processes before, and we're going to go through it again. So if we think about the fasted state, we go into a negative energy balance, blood glucose drops, insulin drops, and then we get these processes in red that are going to increase our Q, that are going to decrease our Q, like increased lipolysis. We don't have a net uptake of lipid to adipose tissue. We have suppression of de novo lipogenesis, and we have decreased glucose uptake to muscle. We have some processes that do potentially push our Q up a bit, like decreased glycogen synthesis, so we're not locking glucose away, and increased gluconeogenesis, which is providing glucose to the brain and red blood cells. But overall, lipid is released and oxidized, glucose is released from the liver to provide fuel for the brain, but it's not the predominant fuel source. The predominant fuel source in the mouse is lipid. And this all flips around in the fed state. We get high blood glucose, high insulin, suppression of lipolysis, lipid uptake to adipose tissue, de novo lipogenesis, and glucose uptake to muscle will go up. You also have an increase in glycogen synthesis and suppression of gluconeogenesis. But what I would argue as a physiologist is this is all mechanism here. We can just black box this, and we can simply say increased food intake or positive energy balance is what results in your RQ. Everything in the middle converges on these values. Why is this? Well, the reason for that is you are what you eat, okay? Every diet you feed a mouse has a food quotient, okay? And this is the RQ of the oxidation of the diet. It's often calculated using values which are known as Atwater values, and this is the RQ of the metabolizable energy of the diet. And it assumes specific absorption coefficients for the macronutrients. But what this actually means chemically is that every diet has a ratio of carbon to oxygen in it. And if we think about a diet made purely of fat, then the only oxygens in that are the carboxyl groups, are coming from the carboxyl groups. And they're going to have a ratio of carbon to oxygen of about 17 to 1. Okay? Because you're going to have these, if it's triglyceride, you've got these glycerol backbones, something like that, or 8 to 1. Whereas a pure carbohydrate diet is going to have a 1-to-1 ratio of oxygen and carbon. And if we are thinking about our mouse as a black box, our mouse is acting like a candle. It's just burning stuff. So therefore, the amount of oxygen you need to burn all this diet and convert it to carbon dioxide and water is not regulated by processes going on in the animal. The amount of oxygen is regulated by how much carbon and oxygen is in the food, okay? And so conceptually, if our mouse is a candle, its RQ over 24 hours will be exactly the same as the food quotient. It's just burning this food and turning it into carbon dioxide and water. However, this assumes that mice are in neutral energy balance. Now, we've seen from our previous talks, mice are almost never in neutral energy balance. So we saw this last time, and this is a relationship between weight loss, delta body weight, and food intake. This is, again, the stunning result that if you do not eat, you lose weight, okay? However, we can also see if you do not eat, your respiratory quotient goes down. So there's a respiratory quotient on the y-axis, food intake on the x-axis. We have a high-fat diet in purple and a chow diet in blue. And you can see these lines are sort of regressing down. Sorry, the intercepts here are wrong. Are regressing down to somewhere around about 0.74, 0.75. And you can never get a respiratory quotient that's truly down to 0.7, except in states that are ketotic, because red blood cells of the brain are reliant on running on non-lipid energy sources, either carbohydrates or ketones. So the point behind it is what's going on here is if you're feeding your mice a diet with a food quotient of 0.91, but they aren't eating it, they still need the energy because we know it's a zero-sum game. You've got to have the energy. So where are they getting the missing calories from that aren't in their food? Predominantly, they'll get it from their fat reserves, and burning fat has an RQ of 0.7. This is certainly true in mice, and it happens very acutely in mice. Within about 4 hours of not eating, they're going to switch to using fat. Humans have the capacity to use glycogen and for much, much longer. Okay. So now we get into a concept, which is if we regress change in body weight against respiratory quotient, if you remember I said if a mouse was not changing its body weight, it was in neutral energy balance, then its RQ should be equivalent to its food quotient. Well, conceptually, if we regress change in body weight against respiratory quotient, The intercept at no change in body weight should be the food quotient. So this is some data from that we had from, I think, DMC, this data set, which was using a high-fat diet with a food quotient of 0.827, and far too neatly, the intercept of regressing these mice against change in body weight is 0.827. In other words, the respiratory quotient at 0 change in body weight is the same as the food quotient. The chow are pretty good. They regress to an intercept of 0.9, and the food quotient is 0.91. So it's very close. So we would can conclude from this the 24-hour respiratory quotient, once corrected for body weight, is equivalent to food quotient. And therefore, any changes in 24-hour respiratory quotient observed in mice are a product of changes in energy balance, not a product of changes in gene function. Okay? And I have a caveat here, which is that RQ corrected for delta body weight can either exceed or be lower than the food quotient under 2 specific circumstances, and that is nutrient malabsorption. So you've got a problem with your guts. So actually, the food that gets into the mouse is not the same as the food it eats. And so if there's a difference between your groups in, say, lipid absorption, that will drive respiratory quotient up in the animals with poor lipid absorption. You could also have non-metabolized elimination of nutrients, e.g. glucose loss in urine, will drop your respiratory quotient down because what's being oxidized by the mouse has less carbohydrate in it than what it ate because it lost some in urine. So is there empirical evidence to support this claim that respiratory quotient just averaged up over 24 hours is essentially just something you can measure with some weighing scales and a diet sheet? So if this is true, we should be able to massively mess up a process that affects lipid or carbohydrate metabolism without changing 24-hour RQ once we've corrected for the energy balance. And so I would argue, and what we're going to look at today is lipolysis is pretty important as a process, okay? Because we do a lot of lipolysis as mice. So we're going to delete ATGL. So ATGL is adipose triglyceride lipase, is an enzyme that catalyzes the hydrolysis of triglyceride to DAG in multiple organs, particularly in adipose tissue. And loss of ATGL will render mice so defective at releasing stored lipid that if you lack ATGL, you will die within 4 hours as a mouse if you are fasted. And that's about the length of time it takes to deplete all your glycogen stores. So you basically deplete all your glycogen stores and then you're out of nutrients and you will die. These mice are extremely defective in mobilizing lipid from their adipose tissue. And we can see here, this is a mouse on a chow diet, and it's looking at its respiratory quotient profile over 48 hours. And what we can see is despite this diet having a food quotient of above 0.9, at this stage here, we're observing, and this stage here is basically in the morning when the mice have gone to sleep, they're using maybe 70% of their calories that they are expending are coming from stored lipid. And to get those calories out of the stored lipid, you need ATGL. So we're going to completely stuff up a large swathe of this plot. And then because we can't do these low RQ values, we're going to shove our RQ up. Or if what I'm arguing is true, we're not going to see anything. And the answer is we see nothing. Knocking out ATGL, even though we can no longer do lipolysis, does not affect the 24-hour RQ. Okay, so I think I would argue we've established RQ averaged over the full 24-hour circadian cycle is correlated with body weight and basically dependent on the respiratory quotient of the nutrient intake. You're welcome to agree or disagree. As I mentioned, we can obtain this information with the info sheet from our diet manufacturer and a set of weighing scales. So the question is then, is there any value to RQ that we can get beyond that? And this is the critical point. RQ fluctuates over 24 hours. We've seen that in a previous slide. It does not run at the food quotient of the diet the whole time. So we can range from 70% lipid utilization on the same diet to, in this case, we've got RQ values over 1. And so these RQ values over 1 are like pure carbohydrate oxidation plus a significant component of DNL, which has this RQ of 5, which is driving our respiratory quotient above 1. So we have this huge range in nutrients. So you can go from 100% carbs plus DNL to 30% carbs, 70% lipid oxidation down here. So it's definitely the case that there's a vast range in what can be done. So is this range important? Does it tell us anything? The importance of our Q range. I'm going to argue, yes, it very much does. So what I've drawn here is some theoretical respiratory quotient profiles. And what we have is in green, a mouse which is able to do a lot of different types of metabolism at different times. So it's good at doing carbohydrate oxidation, good at doing de novo lipogenesis, and it's also good at turning off carbohydrate metabolism and doing lipid release and lipid oxidation in the day phase. And then the red line is a mouse which just can't do it at all, and that just has an RQ for the whole 24 hours that are exactly the same as its food quotient. The orange represents a somewhat intermediate state. So they all have, because I've drawn them, exactly the same average respiratory quotient. This area here and this area here are the same. But I would argue that the metabolism of the mice is radically different in these animals at different times. Now, I would argue that the amplitude of this respiratory quotient graph could be considered a measure of metabolic flexibility. How flexible is the animal at switching between carbohydrate and lipid utilization? Now, this is not an idea I've come up with. It is a long-standing concept in the field of metabolism, and I really think it was first popularized by David Kelly. And so what David Kelly did was he defined metabolic flexibility as the ability to make the transition between using carbohydrate to using lipid and vice versa. And this process occurs on a daily basis in humans. At night, if we're healthy, we should be doing a pretty decent chunk of lipid metabolism. And then after we eat a meal, we switch to doing pretty much carbohydrate metabolism. And so some of their earliest studies they did on this involved doing experiments looking at the respiratory quotient across a leg. And the way you do this is you put catheters into the artery and catheters into the vein of the leg. And you then measure the oxygen and carbon dioxide concentrations in the artery and the oxygen and carbon dioxide concentrations in the vein, leaving the muscle. And from that, you can calculate how much oxygen was consumed and how much CO2 was produced, and therefore you can calculate your respiratory quotient. Now, what they did was they compared lean, healthy individuals and obese individuals with weight loss. And they didn't use a fasted and a fed state, they used a true fasted state, but then they clamped the individuals to create a sort of a pseudo-fed state. So they pumped them with insulin, and then they gave them exogenous glucose to prevent them going hypoglycemic and dying. And the lean, healthy individuals started in the fasting state with an RQ of about 0.84, and then they went up to pretty much 1 in the insulin-stimulated state. So they went to basically using pure carbs. Obese individuals who presumably had a degree of insulin resistance, in the insulin-stimulated state, they showed a degree of insulin resistance and they had less carbohydrate utilization. But I think that's not that surprising a result. What I think is more surprising is what's here. In the fasted state, obese individuals were over-reliant on carbohydrate as a fuel source. So what's going on here? What is it about being obese that means you end up using too much carbs? Oh, and we can think about this range being delta RQ, or your degree of metabolic flexibility. So there are 2 major organs implicated in metabolic flexibility, I would say, in the literature. I think it's going to be the case that it turns out most organs have some role in it. But the one I'm going to focus on first is adipose tissue. You probably already know this, given the institute you're working in, but there are 2 forms of adipose tissue. There's brown adipose tissue, we mentioned it already. It's a thermogenic organ that converts energy into heat. And then there is white adipose tissue, which is the body's main energy store. And I would argue it has 3 main functions. This is why the slide is here. I'm not really telling you about what adipose tissue is, because I think the first 2 get a lot of attention and the third gets considerably less. So the first is the body's long-term nutrient store that can store months of energy so it can get you through times of lean and prevent you from dying if there's a famine or something like that. The second, which is many people say, oh, and adipose, with the discovery of leptin, adipose tissue ceased to be considered as this inert lump of fat and now became recognized as an important endocrine organ. And I've said here it secretes over 50 different hormones and cytokines. I wouldn't be surprised if this is much near 100 now. And the third one, which I think gets less attention, is it also acts as the body's lipid buffer. And I would argue the following, which is that adipose tissue is pretty much analogous to the liver, in that the liver stores excess glucose from our diet, excess carbohydrate, as glycogen when we eat. And then when we stop eating, when we go into a fasted state, like when we're asleep, it's able to release that glucose again when we need it. And I would argue adipose tissue does the same thing over the same time frame, over a daily cycle. So when we eat, if we've eaten more fat than we need, it stores the free fatty acids as triglycerides and releases them when they are needed, again, say when we're asleep in the fasted state. And this process is dynamic, and we can think of glycogen being analogous to triglyceride and glucose and free fatty acids being analogous. And what's very interesting is that these processes are messed up in obesity. Okay, so this is another AV difference experiment. It's from Keith Frain's laboratory in Oxford, and it's some lovely work done by Siobhan McQuaid back in 2012. It's not the first time they made this observation, but I like this graph a huge amount. And what it shows is lean, healthy individuals in black and obese individuals in white. And what we're looking at here is the flux, the release or uptake of fatty acids across a adipose tissue depot. And so this is, again, AV difference. You have the artery catheter, you have the venous catheter leaving the adipose tissue, and you measure how much free fatty acids were in the artery and how much left in the vein, and use tracers to get a more accurate measure of this. Anyhow, what it shows is that in a lean, healthy individual, when we start the experiment, they are releasing lots and lots of fatty acids. If it's negative, it's release. They're releasing lots of fatty acids, and then they eat, and very rapidly they start taking up and storing fatty acids. After the first meal, they peak, comes back down, goes back up, comes back down, goes back up after dinner, comes back down, and then we go back into a releasing phase during the night when the individuals go back to sleep. Now, fatty acid uptake into adipose tissue, much like glucose uptake into adipose tissue or muscle, is insulin dependent. The obese individuals who have a degree of insulin resistance have impaired lipid uptake into adipose tissue. I think that's pretty unexpected. But we also hear obesity is a disease of high lipolysis, but actually that may not be entirely true, certainly not on a tissue level, on an individual adipocyte level, because these obese individuals actually have low free fatty acid release in the fasted state, and they do not have greater amounts of free fatty acid release following the end of the protocol. And what you can see here is this is starting to look very much like the red line of the RER profiles in our theoretical mice RER profiles I drew out is the obese ones, and the healthy ones have a nice flexible range, a big amplitude in their free fatty acid release. So which other organs and processes regulate metabolic flexibility? I talked already about muscle. I would argue actually metabolic flexibility started life and is to an extent still a pretty musculocentric field. And the processes that have been really described to be important are related around the capacity for mitochondrial oxidation of lipids. And there are lots of molecules that have been implicated. I'm sure there's a lot more now. As I say, this is quite an old slide. But overall, total mitochondrial capacity, so things like mitochondrial DNA levels, are correlated with metabolic flexibility. PGC1 alpha, regulator of mitochondrial function. Carnitine acyl transferase, so that's how you're going to get your fatty acids into the mitochondria. UCP3, it's a mitochondrial protein and a putative uncoupler. And acyl carnitines. And so the loss of the actions of these molecules directly reduces lipid oxidation, but is also suggested to lead to the buildup of toxic metabolites So intermediates really in the oxidative process, which don't get all the way, such as acylcarnitines themselves, and lipids like diacylglycerides, so incompletely oxidized fatty acid, triglycerides, and this causes insulin resistance, and that's what blocks your glucose uptake in the fed state. Just a side note here, it has been suggested from clamp studies that the main effect on muscle in terms of glucose metabolism is actually at the level of uptake rather than the downstream metabolism. So there is a bunch of people who say, actually, you get less glucose oxidation in a clamp setting just purely because of transport. There's no actual problem with the oxidation in the mitochondria. So I mentioned that as an aside. Okay, so can we translate all of this stuff from humans, which is done with these AV differences, which are, I would argue, virtually, if not actually impossible in a mouse? Can we detect these changes in animals? And so what I would argue is because mice have this huge range of respiratory quotient, so in a healthy one from 1.1, perhaps up here, down to 0.74, we can just use the amplitude of their free-living respiratory quotient as a measure of metabolic flexibility, okay? And because we're feeding them the same diet and we've got a lot of other control things, I think this will work quite well. And to give you an example of how much this amplitude can be affected, green versus red, we can look at what happens with a mouse with no fat. So if you have a mouse with no fat, you lose all flexibility. You lose all amplitude effectively in your respiratory quotient. This is just 2 mice. I haven't averaged anything. I've just literally gone with 2 mice from the calorimeter. And you can see here, 48-hour cycle, red is wild type, and you can see the wild type's doing a nice process whereby it increases its RQ during the dark when it's eating, and it comes right down to nearly 0.7 in the light phase. And it does that twice. The AZIP mouse, which has no fat, just carries along at the same level pretty much for the whole time. This spike up here, by the way, was the cage being open to weigh the animals, okay? And the reason for this is there's no buffering capacity coming from the adipose tissues. So when the mice eat their food, they can't store their fat, because what these wild-type mice are doing here, they're eating food which maybe has a food quotient of about 0.994, something like here, but they're storing all the lipid in the adipose tissue and just burning the carbs. Well, the AZIPs have no adipose tissue. They can't store it. And then when we get to this point, the wild-type mice are going to sleep, and they are not eating, and they are releasing their fat that they stored earlier and using that as a fuel. Well, the AZIP mice don't have any fat stores, and so they just carry along pretty much the same respiratory quotient the whole time. And it's interesting to note that the AZIP mice are also hyperphagic, and they're almost certainly continuing to eat in these periods, given that they only have about 4 hours of glycogen. So they have messed up feeding patterns. And I've always wondered if this could be an example of the tail wagging the dog. So rather than the brain controlling the feeding patterns, it's actually derived from the peripheral metabolism, which is the key factor in controlling when and how much these animals are eating. Okay, so this is pretty cool because we had this kind of extreme example I just came up with, which was of a wild type mouse in green and then an AZIP mouse in red. That's literally, we can get a biological example of an animal with very, very nice metabolic flexibility and a big amplitude of respiratory quotient. And we can also get a biological example of one with absolutely no ability to fluctuate its respiratory quotient. But what about the intermediate models of impaired metabolic flexibility, right? So where we're not looking at mice as extreme as the AZIP, it's not very hard to tell that those are sick, but looking at mice with more subtle impairments, what can we learn from metabolic flexibility in these cases? First thing is we need to define metabolic flexibility. And for my purposes, I've used the interdecile range. And there's a few reasons for this, which I'm not going to go into at this point. I'm just telling you what the definition is. But it's essentially the highest 10%, the lowest 10%, you subtract the low from the high, that is your metabolic flexibility range. So the first gene I'm going to talk about implicated in this is PPAR gamma. And PPAR gamma is a regulator of metabolic flexibility in adipose tissue. So peroxisome proliferator activated receptor gamma, PPAR gamma, is a key transcription factor. It's necessary for adipogenesis, and it has two isoforms that differ on the protein level. Gamma-1 is broadly expressed and is implicated in loads of different functions. It's necessary for embryonic development. It's implicated in adipogenesis. It's implicated in inflammation, thermogenesis, and control of insulin sensitivity. PPAR gamma-2 is more adipogenic than gamma-1, and it's also known to be nutritionally regulated, which is really at odds at processes like embryonic development and adipogenesis. And by nutritionally regulated, I mean if you fast a mouse, its levels of gamma-1 remain pretty static, gamma-2 goes down. If you overfeed a mouse, gamma-2 goes through the roof, gamma-1 remains pretty static. So this suggests a more dynamic role for PPAR gamma than just long-term maintenance processes like adipogenesis. And one of the nice things for us is that the expression of PPAR gamma-2 is massively enriched in white fat compared to other tissues, whereas PPAR gamma-1 is broadly expressed. So if we study a gamma-2 knockout, we're really looking at main effect of adipose tissue. So the first thing to note is that mice lacking PPAR gamma-2 are somewhat metabolically inflexible. It's nothing like as extreme as the AZIT. We do have this sort of circadian curve, but I would argue this is like the orange line. It's partially impaired. Okay, and the delta RQ for the wild types is shown here in blue, and then the delta RQ for the gamma-2s is compressed. This is the really interesting thing. So these mice are insulin resistant and metabolically inflexible, and if you measure insulin resistance with an ITT, they are, but their glucose tolerance is normal. Okay, so the question is, why would their glucose tolerance be normal, given that we think metabolic inflexibility is associated with insulin resistance and obesity, and we know those are processes, at least in humans, associated ultimately with glucose intolerance and progression to diabetes. In fact, if anything, if we look at 10 minutes, the PPAR gamma-2 knockout has improved glucose tolerance. Okay, so what's going on here? So the point behind it is we do our GTTs in the morning, and we do it at a period of time when the PPAR gamma-2 mice in white are primed to use more glucose than they should. So in a sense, we have an animal which we have rendered metabolically inflexible, and that means when we're doing our GTT in a sort of a fasted or pseudo-fasted state, and we give them the glucose, we're giving it at a time when the animal's metabolism is programmed to overuse glucose. So you give them a load of glucose and they're like, bang, I'll take that up and I will use it. So what about lipid metabolism? So if we look at the RER profile of the PPAR gamma-2 mice, we can also see, in addition to this elevated respiratory quotient in the morning, in the middle of the night, when the animals should be using carbohydrate as almost their exclusive fuel source, there's a somewhat lower respiratory quotient. And this could just be a direct effect, increased lipid usage driven by some effect happening in muscle or brown fat, for example. But PPAR gamma-2, in conjunction with PGCs, is known to actually promote lipid oxidation. However, what's much more likely, given PPAR gamma-2's role in white adipose tissue, is impaired storage. So can we test the impaired storage, or if the gamma-2 mice have impaired storage? The way we do this is we just challenge them with a lot of lipid all at once and see what they can do in the way of storage. And if we give them an oral gavage of lipid, in this case 200 microliters of olive oil, what you see is that there is a spectacular impairment in lipid clearance in the PPAR gamma-2 knockout mice. And just note, compared to a GTT, we're working over a time frame here of 8 hours. You can get away with 6 if it's just wild types, they're back to baseline. The gamma-2s take about 8 hours to return their lipid levels to normal. Okay, so they're severely impaired in their capacity to clear lipid. So what we'd argue here is removal of a key transcription factor in adipose tissue that controls adipocyte function has a limited or no effect on the GTT. It impairs metabolic flexibility and it inhibits lipid clearance. However, if we only had impaired lipid clearance as a source of metabolic inflexibility, and that wasn't partnered with impaired release, impaired lipolysis, adipose tissue would soon cease to exist. And we know the gamma-2 has an impairment in lipolysis, but there's an interesting question here. If we, as a primary effect, block lipolysis, what does that do to our metabolic flexibility? So we go back to the ATGL mouse. I mentioned this earlier. This is the mouse that lacks this key lipase, which is necessary for liberating fatty acids from triglycerides. And a loss of ATGL renders mice so defective at releasing stored lipid that they will die in 4 hours once their glycogen runs out. Okay? We already showed that their average RQ over the 24-hour period was the same, but what about their metabolic flexibility? Well, if we pass into low and high RQs, the lower RQs of these mice, the low 10%, are massively higher. The ATGL knockouts, I'm sorry, are on the left in red, the wild types are on the right. And although it doesn't quite reach statistical significance as a P of 0.07, their higher values tend to be lower as well. So even though all we've done is hit a lipase, there seems to be some crosstalk between the process of lipid uptake and lipid release. And if we impair either uptake, clearance, or we impair lipid release, then we will mess up metabolic flexibility. Okay, so far we have seen that an AZIP mouse with no fat has essentially no metabolic flexibility. That's fine. And that mice lacking PPAR gamma 2 have impaired lipid clearance and impaired metabolic flexibility, whereas mice lacking ATGL have severely impaired lipolysis and also impaired metabolic flexibility. So these are all adipocentric models. No fat, loss of adipose triglyceride lipase, loss of adipocyte-specific transcription factor, PPAR gamma 2. And that partly comes from my background. I'm largely an adipose tissue biologist. But then it leads to an interesting question. Is metabolic flexibility solely a phenomenon of AT dysfunction in mice? And I'm going to now talk about a more generalized example, and this is the AKT2 knockout mouse. Okay, so AKT2, I think most of you will be familiar with, it's also known as PKB. It is a critical node in the insulin signaling pathway. It's downstream of the insulin receptor, downstream of PI3 kinase, and there are three forms of AKT, 1, 2, and 3. It is generally thought that AKT2 is the most important for controlling metabolism, metabolic nutrient utilization. Whereas AKT1, for example, is more important in cell growth. AKT2 is really the critical one for glucose uptake. I am certain it's not that neat and there's crossovers between, but that certainly, I would say, is the broad picture idea. And there have been many AKT2 knockout mice, but there were two separate models first made in the late 90s, early 2000s, and they were phenotypically distinct. The Birnbaum mouse was mildly glucose intolerant, it had a loss of fat mass, and it was hyperinsulinemic. Okay? The Coleman mouse had all of the above, but it also had a loss of beta cell mass and frank diabetes. And this was, as is always the case when there are divergent phenotypes in mice, put down to genetic background differences. In this case, I think it's probably true, because I once crossed an OBOB onto an FBBN background, and we got some of these OBOBs just went to complete frank diabetes. So not like having a bit more water and a bit more urine in the cages, they were actually started to lose so much weight we had to kill them, and they were drinking 50 mils of water a day. So this is certainly possible that a genetic background can cause frank diabetes in one setting and not in the other. We have the Birnbaum mouse, so this milder phenotype. So what are the effects of AKT2 ablation? So first of all, we have greatly reduced fat mass on both chow and high-fat diets. Now, I didn't mention it and show the data, but the gamma-2 has very, very little change in fat mass. The AZIP has no fat mass. The AKT2 knockout seems to sit somewhat in between. They also have a bit of a loss in liver weight and generally in lean mass, okay? So this is wild type on chow, knockout on chow, wild type on a high-fat diet, knockout on a high-fat diet. These are the males. But you can see huge effects on subcutaneous white fat, BAT, and EWAT masses, okay? So these mice are partially lipodystrophic. They also have severely impaired metabolic flexibility on a chow or a high-fat diet. And this effect is somewhat larger than gamma-2 and somewhat less than the AZIP. So this is about a 30% reduction on chow in metabolic flexibility. High-fat diet just suppresses metabolic flexibility as a function of the diet, because although the RQs don't drop below 0.7, the chow gets down to about 0.7374 usually. In the case of the high-fat diet, it can also only get down to about 0.7374, but it never reaches those high values. It never gets up to 1.05 because they're never going to do any DNL. This is a 60% high-fat diet, so they really only get up to about 0.8 at most on this diet. So you've got really, or 0.84 maybe. So you've really got this suppression of the potential range of metabolic flexibility. But again, we see about a 20 to 30% reduction in delta RQ as a result of our AKT2 knockout. So what are the consequences of impaired MET flex and in a mouse model of generalized insulin resistance, congenic insulin resistance. So we don't unfortunately have all the nice data. This is data we did in collaboration with other people. They had specific experiments they were interested in doing. But what we do know is these mice are congenitally insulin resistant. We don't have the lipid tolerance test, but we are able to look at glycemia. We know whether they're hyperglycemic, and we can also look at the fed fasting transition in these mice because we looked at glycemia in the fasted state and the fed state. So the first question is, are they hyperglycemic? Now in humans, one of the definitions of diabetes is if you have fasting hyperglycemia. So these mice are severely insulin resistant. Do they have any indication that they may be pre-diabetic with, say, impaired fasting glucose? Well, let's have a look. The answer is no. Rather bizarrely, they actually have improved fasted glucose on a high-fat diet and normal fasting glucose on a chow. And this result has been seen before. This is Morris Birnbaum has published this result. And so why is this? Well, one argument is, and I think that's the argument in the literature, is that they just over-secrete insulin. But it's never been very satisfactory to me why they would do this. But I think if we can think about it less from a insulin as a controller of the blood glucose levels model, but more from a metabolic flexibility model, it makes a bit more sense. Because when we look at the low 10% of the respiratory quotient values in these mice, they are higher. They are elevated in mice which are AKT2 knockouts. So they have elevated respiratory quotient, and therefore that suggests they have an inappropriately high glucose utilization in either the pseudo-fasted or fasted states. Now, what happens in the fed state? This is a bit different. There is profound hyperglycemia in the fed state. So what's going on here? Why is this different to the fasted state? So in the fasted state, we may be in a situation where, because we've messed up lipid uptake into the adipose tissue, we've probably messed up release as well. Otherwise, we would be losing lots of fat. Now, I know they have lower fat mass, but the mice have massive turnover rates of fat. So it has to remain relatively imbalanced. So if you put in a mixed meal, and a mixed meal is a combination of carbs and fat, into the system, you will get a competition between the carbs and the fat as substrates. If the fat is not being locked up properly in adipose tissue, you get two things. You get an overutilization of lipid in organs like muscle, and this will also result in a failure to appropriately take up glucose. Now, these mice are also insulin resistant in the liver, and they may have problems with doing DNL. All of this adds up to having too much glucose sloshing around the system. And so we get these very high fed glucose levels on chow and on high-fat diet, up to 30 millimoles. And again, we go back to this. This makes sense from the point of view of RQ, because the knockouts have essentially got a lower RQ in the fed state, the high 10% of values, suggesting they aren't using the glucose and it's building up in blood. So these glucoses are really high, okay? So could it just be diabetes or some equivalent? You've got some beta cell failure going on. Maybe the Birnbaum mouse has problems with its beta cells in the same way the Coleman mouse had, but not as severe. Well, I would argue that the data we have is not that compatible with that hypothesis, because what you would expect to see then is a huge degree of variability in the blood glucoses, because you would expect some mice to be further down the track to beta cell failure than others. And so this very high value of 30 would be made up of mice with 60 and mice with 12, like wild types. But that's very much not the case. The variation is not that much greater. The blood glucose is not uncontrolled. We can see here individual knockout mice, and they vary a bit more than the wild types, but it's not spectacular. And the same is also pretty much true of chow. We've got a bit of an outlier here, but they don't vary as much. This is a subset of the animals. The actual data you were seeing earlier is N of 8 and 8. Okay, right. So how does this translate into glucose tolerance tests? Okay, so we know we've got this profound insulin-resistant mouse. Okay, so this is the last bit of the talk, so don't worry, we're ending soon. Profoundly insulin-resistant mouse. And we're going to go do our metabolic tests that we do on all our mice to check whether they're insulin resistant. We're going to go do a GTT. But we saw earlier when we were looking at the PPR gamma 2 mouse, which is another model of metabolic inflexibility, like the AKT2, that they were able to pass the GTT because this concept that when we do them fasted overnight, in the morning when the mouse is programmed to overuse glucose, then we give them some glucose and they go, thank you very much, and they clear it perfectly well, if not better. But can this hold true in the AKT2 knockout mouse, a model of congenic insulin resistance? And the answer is going to be yes, it can clear it. And what we see here is that the wild type and the AKT2 knockout mice have the same GTT area of curve. Slightly different shapes to the profile. The knockout started off with a lower fasting glucose. They never reach the same peak, and then they slightly slide out here, but they certainly don't reach statistical significance for the areas of curves. Now, this surprised me immensely when I did this, because I went to the literature and I had a look, and there were 8 papers that show impaired glucose tolerance in an AKT2 knockout. But I was using our standard protocol at the time, which is an IP of 1 gram per kilo of glucose. When I looked at all 8 of those papers, every single one had used at least 2 grams, and in some cases 3 and 3.5 grams of glucose. So I think what might have been going on is all these researchers had done their 1 gram per kilo, or at least a good few of them, gone, our AKT2 knockout mouse has normal glucose tolerance. And then they thought, let's give it more glucose. And that may well work, because if you give it enough glucose, you're going to get a stronger insulopenic effect, and insulin may become more important for this curve. But it's certainly the case at 1 gram per kilo, you are getting no difference in the GTT between a wild type and an AKT2 knockout mouse congenically insulin resistant. And I would argue that whilst you could say, okay, well, your challenge is too weak, it's only 1 gram per kilo, you should do 2 or 3 to get a difference, I would argue it's not that weak. Because if we look at a wild type, this dose of glucose we're using is sufficient to boost our glucose levels in blood to double that of either the fed or the fasted state. And I think it's even more remarkable when we look at the AKT2 knockout in the fasted and the fed state, because what we see is same dose of glucose is not even able to get the glucose levels in the blood of an AKT2 knockout mouse to reach that of an AKT2 knockout that is just randomly fed, ad libitum fed. So, well, what I would suggest is the case here is what I've alluded to earlier, that when we do our GTT, it is a mononutrient challenge. We are just giving glucose, and we're giving it into a state where the animal is overly reliant on glucose and is really primed to use it. Whereas when we're looking at the fed state, we have a mixture of lipid and glucose, and these things are competing. And also, the other point is these animals will be in positive energy balance. So there'll be more nutrients going in that the organism can handle. Okay, so summary, and we're coming to the end. Fed blood glucose levels are as stable as fasted blood glucose levels. Despite having normal glucose tolerance when compared to wild-type controls, AKT2 knockout mice are insulin resistant by definition and have more than double the fed blood glucose levels of wild-type mice. So conclusion would be that a primary failure to appropriate store lipids to store lipid could inhibit carbohydrate utilization, even in settings where we've got relatively low levels of carbohydrate available for metabolic oxidation. So even on the high-fat diet, that mixed meal effect is screwing things up. So the overall summary. So we've looked at 2 separate models. I think we've looked at 4 in total, but 2 we've looked at in detail, which is the AKT2 knockouts and the PPAR gamma 2 knockout of impaired metabolic flexibility. And so despite the presence of insulin resistance in both these animals, glucose tolerance in the GTT is largely normal, at least at a 1 gram per kilo dose. Both models are characterized by reduced adipose tissue function, either mass in the AKT or direct measures of adipose tissue function in the case of gamma 2, lipid clearance. And both models are characterized by impaired metabolic flexibility as measured by calorimetry. And so I'd argue that there are some implications and future directions you should think about in your own work. I'm not saying you should do these things, but I think you should certainly think about them. And I made this slide many years ago, and since then I've actually tried this stuff out. And you may see in later talks that I think all of these statements are going to be true. So the first question is, should we consider doing GTTs and LTTs in fed states as well as fasted states? And I would argue definitely yes. The gamma-2 mice, whilst having a pretty much normal GTT in the fasted state, actually separate really quite nicely in the fed state. Could mixed meal tolerance tests provide a better readout of whole organism nutrient handling than, say, GTTs? And I agree that's the case. And there's a couple of ways to do this, which is you can either fast your mice, then give them some food back and just measure insulin and glucose over time. And you alternatively can do a diet challenge that I developed, which was a feeding a mice a high-fat diet for one day. I mean, I didn't really develop it. I just started looking at what was happening in the mice after one day of high-fat diet, so calling them. And what you see with both these challenges is that they can discriminate wild type and knockouts, which have normal GTTs, really quite powerfully. You can get 2-3-fold differences, even 10-fold with one day high-fat diet on insulin levels in mice like the gamma-2. So could reductions in metabolic flexibility be a more sensitive assay to detect carbohydrate metabolism defects than GTTs in the screen? And again, we've done this. We're in the process of finishing this up, but we've gone to the IMPC. We've got all of their mice, got the metabolic flex data, got it computed in collaboration with Alexander Banks, and we pull up lots of nice models, some of which are published, and they validate out that this is picking up animals with problems in insulin sensitivity and problems with lipid handling in adipose tissue, but also some are unpublished, and we're working on a couple of them. And could they in particular pick up animals with defective adipose tissue? I would say we're not quite there. We need to work out which tissues are overrepresented in animals with impaired metabolic flexibility, where the genes are expressed. So we think it could be the case, but we're at the moment in the process of trying to confirm that. Okay, so that's it. So thank you very much. And now I will be happy to take questions.